Hi, this is Gina Piscatelli with another lecture on the digestive system for Anatomy and Physiology 2 at Madison Area Technical College. This lecture is on the accessory digestive organs, including the liver, the gallbladder, and the pancreas. First, let's look at some anatomy and the structural relationships between the liver, the gallbladder, the pancreas, and the duodenum. So the liver, you can see, is a very large organ, and it creates bile, which travels in these ducts called bile ducts to reach the common bile duct. The gallbladder is embedded in the right lobe of the liver, and it stores excess bile. It releases the bile through the cystic duct, which joins the common bile duct, and travels posteriorly to the duodenum, where there's an opening into the duodenum, and a sphincter is located there, called the hepatopancreatic sphincter, or the sphincter of Oddi, if you've ever heard of that. The pancreas, as you know, is under the stomach, and the stomach sits over here, and it has a duct called the pancreatic duct that also leads to the duodenum, and its opening is located in the same place as the common bile duct into the duodenum. And so its contents, the pancreatic duct contents, are also controlled, or the release of them into the duodenum is controlled by this hepatopancreatic sphincter. So the liver, the gallbladder, and the pancreas make secretions or release secretions that enter into the first part of the small intestine called the duodenum. Now, as I mentioned before, we're going to kind of look at functions here. As I mentioned before, the liver makes the bile. And this bile is important because it neutralizes the chyme coming in to the small intestine from the stomach. And you know that chyme is very acidic. It also aids in lipid digestion. So the bile is needed to break down the lipids that enter the small intestine. Now the liver continuously makes this bile, but when the common bile duct is full of bile, it kind of backs up and is stored for when it's needed in the gallbladder. So the gallbladder stores excess bile and releases it when we eat lipids in particular. The pancreas makes pancreatic juice, and the pancreatic juice also neutralizes the acidic chyme, but it aids in digestion. In fact, the most chemical digestion occurs in the duodenum due to these pancreatic enzymes. There are some enzymes that help with lipid digestion, some that help with protein digestion, others that help with carbohydrate digestion, and there's even some for nucleic acid breakdown. So both bile and the pancreatic juice will function in the duodenum, even though they're made in other places by other organs. So that's an overview of function. Now let's look at in detail at the liver, the gross anatomy of the liver. The liver is the largest gland in the body, and it weighs about 3 pounds in an adult. It sits under the diaphragm in the right upper quadrant of the abdomen. It has 4 lobes separated by fissures, And you can see one fissure here on this anterior view between the right lobe and the left lobe. A mesentery called the falciform ligament is also located between these right and left lobes, and you can see remnants of that here. And it suspends the liver from the diaphragm and the abdominal wall. On the posterior side of the liver, and kind of inferior as well, the gallbladder rests in a recess of the right lobe, right here. And also visible are the other two lobes of the liver, the caudate lobe and the quadrate lobe. Now I know you think and know that caudate means towards the tail, so you would think that would be the most inferior. But it turns out that the quadrate lobe is actually more inferior. And they're very small lobes. Also notice on this posterior view, this dark region right here. That's where the inferior vena cava was located before it was removed. Now I'd like to talk a little bit about the vasculature related to the liver. So the inferior vena cava, as we know, lies on the posterior side. And the aorta actually lies more medial to the liver. And off of the aorta, there's a branch called the hepatic artery. It actually comes off of something called the celiac trunk, but the hepatic artery is what supplies arterial blood, which is oxygen rich, to the liver. And it is shown in this diagram in red. It's just very small. Venous blood is drained from the liver by hepatic veins, which will then empty into the inferior vena cava. There are many hepatic veins throughout the lobes, and they'll merge and then eventually dump the blood into the inferior vena cava so that it can travel back to the heart. But the liver has another blood supply called the hepatic portal circulation, or as noted in your textbook, the enterohepatic circulation, but we know it as the hepatic portal circulation. In this circulation, venous blood that's being drained from the small intestine travels to the liver via the hepatic portal vein. So the hepatic portal vein is shown here close to where the hepatic artery is. And this blood has come from the small intestine, where nutrients are absorbed. So this blood is nutrient rich, traveling in the hepatic portal vein, and it provides the liver with glucose so that it can perform glycogen synthesis. It also provides amino acids so that the liver can synthesize plasma proteins like albumin and all the clotting factors that we learned about. For reasons we'll discuss a little bit later, lipids that are absorbed in the small intestine will actually reach the liver through the hepatic artery, and we'll talk about that a little bit later, actually in the next lecture. So this is a picture of the hepatic portal system so that you can see the small intestine where all these amino acids and carbohydrates are absorbed. They enter these veins, which makes sense because blood is being drained through veins, but they merge into a vessel called the hepatic portal vein. And the hepatic portal vein travels behind the stomach to enter the liver and then branches. So there's several branches of the hepatic portal vein that will then travel throughout the lobes of the liver to provide the liver with these amino acids and monosaccharides like glucose. And the hepatic portal veins end up dumping into these huge capillaries, which are called sinusoids. And we'll take a look at those in a little bit. Eventually, though, blood has to be drained from the liver as well and travel back to the heart. And that will happen from the inferior vena cava into the right atrium. Let's look at some microscopic anatomy so that some of this makes a little bit more sense. Lobes of the liver are composed of structural and functional units called liver lobules. And these lobules are six-sided hexagons with a central vein located in the middle. And each lobule is surrounded by a septum made up of connective tissue. So the central vein is actually draining blood towards the hepatic veins that are going back to the inferior vena cava for the regular return to the heart. Now, radiating from the central vein are plates of liver cells, and these liver cells are called hepatocytes. And the hepatocytes are arranged in kind of vertical plates that radiate out. So I've drawn kind of yellow lines around a plate of hepatocytes, right here and up here and there. So that's where the cells are. And all the white spaces in this particular image are where the venous sinusoids are that I mentioned before. These rather large capillaries that are fenestrated, so they're very porous. This diagram might actually help you understand a little bit more about all the blood supply that happens. So let's look at one lobule and talk about the flow of both bile and blood. Okay, so this is our lobule, kind of a hexagon, and part of it has been removed so that you can look into the central vein. Now, blood reaches the liver cells by branching off of the aorta. So the red indicates small vessels, capillaries that are coming from the arterial blood supply. And that blood drains into these sinusoids, which are in between the hepatocytes. The hepatocytes are brown beige color, and the liver sinusoids are blue. Now, also draining into these sinusoids is venous blood coming from the hepatic portal vein, which, as you remember, came from the small intestine. This blood came from the small intestine, so it's nutrient rich, and it travels to the liver and dumps into the sinusoids. So in these sinusoids, you have a mixture of both oxygen-rich blood as well as nutrient-rich blood. Another kind of vessel that we see is a duct that's colored green here, called a bile duct. And these hepatocytes that are located in the liver are making bile. and they secrete the bile into really small vessels called bile canaliculi. And they're kind of offset here superiorly so that you can see that there are these small vessels located in between hepatocytes. They will merge and eventually allow bile to flow into this bile duct. So at the corner of all the corners of a liver lobule is a region that we often call the portal triad. But each triad consists of a bile duct, a branch of the portal vein, and a branch of the hepatic artery. It's only the central vein that will be carrying blood to the heart, back up to the heart, because the central vein connects to the hepatic vein, which eventually merges with the inferior vena cava. So notice that blood and bile flow in opposite directions. The bile moves away from the hepatocytes towards the periphery of the lobule. So it heads in that direction. Whereas the blood moves towards the central vein. So they move in opposite directions. And on this slide, I kind of have written out the direction of blood flow in the liver. So we'll start at the small intestine. Blood leaving the small intestine in veins merge into the hepatic portal vein, which is coming in here. And then the hepatic portal vein branches. Eventually, this nutrient-rich blood enters the liver sinusoids, dropping off all the nutrients, or at least the nutrients that the liver needs, and then it goes into the central vein. And so all these sinusoids are sending blood that came from the small intestine into the central vein. And then the central veins in all the lobules, in all the lobes, will eventually merge into various hepatic veins, which will connect up with the inferior vena cava and return to the heart. Now, bile flows from the liver eventually to the small intestine. But first, of course, it's made in the liver by the hepatocytes, and they secrete the bile into these bile canaliculi, which have ducts that lead out to a hepatic duct. And so we call these lobular bile ducts, these little small ones. And then we get into a hepatic duct. And eventually that will merge into what we have seen in the cat dissection, the common bile duct, which will bypass the gallbladder, I guess, and enter the small intestine, the duodenum in particular. So let's now look at the functions of the liver, particularly just the digestive functions. The liver processes this nutrient-rich blood received from the small intestine by the hepatic portal vein. What it does with the nutrients is really what we would call a metabolic function. So its major digestive function is to secrete bile, and this bile is needed to chemically digest lipids. That's what it's important for. So now we're going to talk about bile. You already know that it's produced by the hepatocytes of the liver, and it will eventually be released into the duodenum of the small intestine. But when there's excess bile, it'll be stored in the gallbladder. Now, what does the bile have in it? You already know a little bit about what's in bile because when we did the circulatory system, we talked about how red blood cells are broken down when they're aged, and the hemoglobin can actually be reused, at least the heme part and the iron part and the globin, the protein part, can be reused. But there's also bilirubin made from the heme, and it breaks down into, well, the bacteria that are present in the small intestine, converted into this kind of yellow product, or I'm sorry, brown product called stercobilin, but it doesn't really matter. We only need to know bilirubin. So in bile, we have bilirubin, which is a byproduct of the breakdown of red blood cells. We also have bile salts, and the hepatocytes make these salts, which are basically ions that help to dissolve cholesterol and other lipids. Also present in the bile are phospholipids, which are really important because they would have both a polar region and a non-polar region, which would really help emulsify lipids. Cholesterol is present in bile, and triglycerides, as well as sodium bicarbonate, which is the agent that can neutralize acidity. The bile salts can be reused after they've been dumped into the small intestine to help dissolve lipids, and I'll show you about that in a little bit. So the function of the bile is to help break down lipids in the small intestine. In addition, a major function is that since this bile is going to be dumped into the small intestine, it will mix with the chyme, and much of it will reach the large intestine where it becomes feces. And so this is a major way that the body can rid itself of cholesterol. So cholesterol made in the liver can actually be excreted from the body. And we'll see later when we do metabolism that cholesterol is actually synthesized from various lipids and some proteins in the liver. So the bilirubin is excreted with the feces. The cholesterol is excreted with the feces, although some is reabsorbed. But the bile salts can be reused. They can be recycled and reused. Before we talk about the bile salts being recycled and how that happens a little bit, let's look more at the function of bile related somewhat to its composition. So that's what I'm going to go to next, just so you know. So when we eat something with fat in it, through mechanical churning of the stomach, as well as segmentation or contraction in the small intestine, generally speaking, the fat is just mechanically broken down into these globules. But it's really not completely chemically digested so that we could absorb it. So what happens is that the bile salts that enter the duodenum will break down this large fat globule into smaller fat droplets. And you'll notice that the bile salts here kind of surround a small portion of the fat globule. So the fat globule gets coated with bile salts. Eventually, the bile salts break down the fat globules into smaller little droplets surrounded by bile salts still, called micelles. And then the micelles are traveling in the small intestine and bump up against the small intestine brush border. Remember we had brush border enzymes on the microvilli of the small intestine wall? Well, there are lipases present on that small intestine wall that will break down a lipid into individual fatty acids and monoglycerides. So if you remember, a triglyceride looks like the letter E, where you have this carbohydrate backbone, I'm drawing C's there, with 3 chains of fatty acids. So the fatty acids are broken off and they can be absorbed across the wall of the small intestine, and a monoglyceride right here can also be absorbed by the small intestine. Once these broken down fats, lipids, triglycerides, whatever you want to call them, are absorbed, they don't enter the hepatic circulation, and instead they enter the lymphatic circulation. So they won't travel in the hepatic portal vein. Instead, they will travel in the lymphatic system, which will, of course, eventually reach the circulatory system, because the lymphatic system connects at the subclavian veins via ducts to the circulatory system. Okay, let's look at the recirculation or recycling of bile salts now. The bile is made in the liver, stored in the gallbladder, released into the duodenum of the small intestine. And in the small intestine, that bile helps break down fats and triglycerides. And so as the contents of the small intestine moves along and gets mixed up with these bile salts, we get smaller and smaller fat droplets that I called micelles. And as the fats or lipid components are absorbed into the hepatic portal vein, so are the bile salts. 95% of bile salts that are traveling in the small intestine will be reabsorbed from the ileum of the small intestine into the hepatic portal vein. And now these bile salts are going to travel through the hepatic portal vein all the way back to the liver, so they can be reused. And the liver will only have to make an extra 5% new bile salts. So they can be released again for the next lipid digestion cycle. Okay, other functions of the liver, as I mentioned, are related to metabolism. The liver regulates the level of glucose in our blood by being able to perform gluconeogenesis. That means when blood glucose is low, it can make glucose from amino acids and fatty acids. And those amino acids will have reached the liver by traveling in the hepatic portal circulation. In addition, the liver can synthesize glycogen. That process is called glycogenesis, and it synthesizes the glycogen from glucose. Glycogen is just a chain of many glucoses. And again, that glucose reaches the liver through the hepatic portal system. So that's how glycogen can be made from glucose in the liver. If blood glucose is low, not only can the liver make new glucose, but it can break down any glycogen that has been stored in the hepatocytes. So that's another metabolic function. And remember, this glucose is needed to be transported in the bloodstream to reach all of our body cells so that the cells can use it to make ATP. In addition, the lobules present in the liver, the lobules were the six-sided kind of region surrounding a central vein, remember? Interspersed with the hepatocytes are special cells called stellate macrophages. And those macrophages can remove debris from the blood that has entered it. Things like bacteria that came from the small intestine, or worn out red blood cells, as well as white blood cells. In addition, urea is created when amino acids are broken down, and that travels in the blood to the liver. And ammonia is produced when amino acids are broken down, and cells that break down these amino acids to make ATP release the ammonia into the blood. And so the blood eventually reaches the liver, usually through the arterial supply rather than the hepatic portal circulation, but that doesn't really matter. But the ammonia is toxic. It's a normal byproduct of breaking down amino acids for energy. The liver makes it less toxic by converting it to urea, and it puts the urea into the blood. And now the urea is going to travel in the bloodstream, eventually which flows to the kidney, and the kidney can actually do something with this urea. Get rid of it, basically. And again, these are nitrogen-containing components. The last thing that I want to mention that the liver does is it stores fat-soluble vitamins like A, D, E, and K. Okay, let's move on from the liver. The liver's complicated, but the gallbladder's not as complicated. So let's look at the structure and function of the gallbladder. In, I think, Greek, maybe it's Latin, gallbladder is cholecyst. So cyst refers to a bladder, and chole refers to gall, so gallbladder. And this cholecyst, or gallbladder, is a muscular sac that might appear green when it's filled with bile. And that's what you can see up in this cadaver picture in the upper right-hand corner. The gallbladder appears green. And the liver is constantly making this bile and dumping it into the small intestine, but sometimes this sphincter is closed. Most of the time, actually. Only after a meal does it relax. And so the bile is not going to go into the small intestine 24 hours a day. And so the excess bile that the liver has made gets stored in the gallbladder. And this bile is not needed immediately, but it gets concentrated in the gallbladder because the gallbladder wall can absorb water and some ions. So it becomes more concentrated. Now, since the gallbladder is a muscular sac, it can contract. It's obviously smooth muscle. And when it contracts, it expels the bile into its little cystic duct, which merges with the common bile duct. And that will eventually flow into the duodenum. And we'll talk about how that's controlled a little bit later. So let's talk about bile release and blockage of bile release. When bile release is blocked, bile pigments, particularly bilirubin, which is in the bile, of course, accumulate in the blood and eventually deposit into the skin. It's a yellow color, and so we call this jaundice. One cause of the bile being blocked is the formation of gallstones, and you can see kind of gray gallstones in the gallbladder. The formation of gallstones is called cholelithiasis, and gallstones are made up of cholesterol that crystallizes, and there's some salts in there too, and it crystallizes due to the fact that there's been excessive water reabsorption by the gallbladder. It's also possible that you could get gallstones due to liver dysfunction because the components of the bile aren't quite right, and so it's kind of dehydrated. The biggest problem, I guess, immediately anyway, with gallstones is that when you eat and the gallbladder contracts, it's going to be really painful if there's gallstones in there. But in addition, a big problem is the blockage that might occur. Sometimes the gallstones are past, but blockage that could occur at the opening to the duodenum. And so you'll get inadequate lipid digestion. And the lipids will be not broken down, travel through your small intestine, and you can get pretty bad diarrhea, which would lead to dehydration as well. But if you don't have lipid breakdown and therefore lipid absorption into the bloodstream, you can't absorb certain vitamins. And these vitamins are A, D, E, and K because they need the lipids to kind of stay soluble in the blood and travel to the liver. And of course, a big problem would be that you're not eliminating bilirubin, and that's why you would get this jaundice or yellow color, because the bilirubin would be absorbed across the wall of the gallbladder into the bloodstream and eventually deposit in the skin. I guess that now there's a technique that's used to break down these gallstones without having to do surgery. Many people end up just having their gallbladder removed. I know plenty of people that have that done, but it would be better to keep the gallbladder and perform what's called lithotripsy. And this is like an ultrasonic vibration that pulverizes the gallstones, so you don't have to either remove gallstones or remove the bladder. Kind of cool. Okay, that's all for the gallbladder for now. And now let's look at the pancreas, its structure and function. So the pancreas function related to digestion is an exocrine function. That's because the pancreas makes pancreatic juice that's released into the pancreatic duct. And the cells in the pancreas that make pancreatic juice are called acini cells or acinar cells, either one, acini cells. Remember when we talked about metabolism back in ANP1 and the endocrine system, we also learned that the pancreas makes hormones, insulin and glucagon, that will be released into the blood versus into the pancreatic duct. So the pancreas has both an exocrine function for digestion and an endocrine function for metabolism or maintaining blood glucose. And they're different cells. The acini cells are the ones that make the pancreatic juice, and the cells in the islets of Langerhans are the ones that make the hormones. Pancreatic juice that's released into the duodenum is able to neutralize acidic chyme as well as promote digestion, chemical digestion, and that's because of its components. So pancreatic juice, like bile, has sodium bicarbonate, and that will neutralize the acidic chyme. And pancreatic juice has enzymes that will digest carbohydrates and nucleic acids, as well as lipids that have already been emulsified into those micelles, those tiny little lipid droplets. These enzymes include pancreatic amylase. Remember, we had an amylase in the oral cavity. The saliva contained salivary amylase. And so this pancreatic amylase functions similarly, but in the duodenum, and it breaks down carbohydrates. There's pancreatic lipase that will break down emulsified lipids, and that was those lipids were emulsified by bile first. And there's a couple of enzymes not as important to us that will break down nucleic acids. In addition, the pancreatic juice contains some Zymogens. These are inactive enzymes. They have to be activated in the small intestine, and all of them will digest polypeptides or protein. So we haven't talked about protein since the stomach. So the pancreatic juice Zymogens are trypsinogen, chymotrypsinogen, and procarboxypeptidase. And all of these need to be activated in the small intestine in order to digest polypeptides. So we're going to look at how that works. So these Zymogens, remember, are coming from the pancreatic juice. And the first Zymogen that gets activated in the small intestine is trypsinogen. And it will be converted into its active form called trypsin by an enzyme of the small intestine. But remember, the small intestine doesn't secrete enzymes into the lumen. Instead, the small intestine keeps its enzymes on that brush border, the microvilli. And one of those brush border enzymes is enteropeptidase. And so when trypsinogen bumps up against the wall of the duodenum, enteropeptidase will convert it into trypsin. That's usually the first thing. Now trypsin will break down proteins, and chymotrypsinogen is a protein. So is procarboxypeptidase. So trypsin is able to convert both of these into their active forms, chymotrypsinogen into chymotrypsin, and procarboxypeptidase into carboxypeptidase. Now, since trypsin is an enzyme that breaks down proteins, and trypsinogen itself is a protein, it turns out that trypsin can break down trypsinogen. So there's this positive feedback mechanism that occurs. Trypsin can cleave trypsinogen into trypsin, its more active form. And I have a diagram to show you this so that maybe visually it'll make more sense to you. So the pancreatic zymogens that have to be activated in the small intestine all are important for polypeptide digestion. And there's 3 of them: trypsinogen, chymotrypsinogen, and procarboxypeptidase. And so they're released into the small intestine. Trypsinogen is kind of getting swirled around in this small intestine, and it bumps up against the brush border that has this enzyme called enteropeptidase. I'm getting tongue-tied. And so trypsinogen gets converted into trypsin. Now trypsin then converts the chymotrypsinogen and procarboxypeptidase into their active forms. Now we have trypsin, carboxypeptidase, and trypsin, all active forms, in the small intestine to break down proteins. And as I mentioned previously, trypsin can actually kind of speed up the process of activating these second and third zymogens, because it can create more of itself, essentially. Trypsin can break down trypsinogen, and so you'll get more protein digestion. That's the main objective. Okay, I wanted to mention that the pancreatic duct and the common bile duct will enter the small intestine at the same location. And notice the sphincter, once again, that's located at the opening for these 2 ducts. It's called the hepatopancreatic sphincter. When this sphincter relaxes, both pancreatic juice and bile will enter the duodenum. When the sphincter is contracted, which happens between meals, bile production continues in the liver, and the bile will back up into the common bile duct, enter the cystic duct, and be stored in the gallbladder. However, the pancreas has an accessory duct that allows continuous release of pancreatic juice into the small intestine, but not a great deal. Notice that this accessory pancreatic duct is rather narrow, but there's no sphincter there. So there's always a little bit of pancreatic juice entering your small intestine. The next thing we're going to do is look at what controls release of pancreatic juice and bile at this location of the sphincter. First, there's neural control of both the gallbladder and the pancreas function. So we have to recall the phases of digestion that we talked about with the stomach. There was a cephalic phase. That's when you're thinking about food. There's a gastric phase when the food bolus enters the stomach. And then there's an intestinal phase when the chyme enters the intestine. In these first 2 phases, the cephalic phase and the gastric phase, there's a neural reflex that stimulates the gallbladder to contract, the pancreatic juice to be secreted more prevalently, and it causes that sphincter to relax. And this neural reflex travels via the vagus nerve to stimulate the gallbladder to contract, the pancreas to secrete more juice, and the sphincter to relax. Notice that there's no neural control of liver function, like bile production or bile release by the liver per se. That's just going to happen continuously. But we can get a huge release of bile by contracting the gallbladder, and that's what the nervous system does. There's also hormonal control, however, and these hormones will control liver as well as gallbladder and pancreatic function. So now we have to think about the last phase of digestion, the intestinal phase. And this is when chyme enters the intestine. The fatty chyme will stimulate the duodenum to produce a hormone called cholecystokinin. And the acidity of the chyme entering the duodenum stimulates it to produce secretin. Both of these hormones will act on the pancreas. Cholecystokinin and secretin will act on the pancreas in different ways. Cholecystokinin, as you can tell by its name, refers to the gallbladder. Hopefully that's going to help you remember that it only acts on the gallbladder. I didn't say that right. The gallbladder responds only to cholecystokinin. That would be the best way to say that. And the liver only responds to secretin. So I'll fix that slide when I post it. I'm so sorry. Let's look at the next one. What their actions are, these two hormones. So the actions of cholecystokinin, remember it is released by the duodenum into the bloodstream in response to lipid in the chyme being present here in the duodenum. So when there's lipid in the chyme present in the duodenum will secrete this hormone and release it into the bloodstream. Now this hormone, cholecystokinin, targets the gallbladder to contract, the pancreatic juice to contain more lipase, and relaxation of the hepatopancreatic sphincter. So that kind of makes sense because you're trying to digest lipids. And so when there's a lot of lipids in the diet, cholecystokinin will be produced by the small intestine, causing the gallbladder to release bile, the pancreas to make more lipase, and for both things to be able to enter the small intestine because that sphincter relaxes. The other hormone, secretin, also made by the duodenum cells, enteroendocrine cells in the duodenum, make this secretin in response to the acidity of chyme. So all the actions are going to be of this hormone are going to be aimed at neutralizing the acidity, bringing the pH up closer to neutral. And the way it works is secretin in the blood reaches the liver, and now the liver, making the bile, will increase how much bicarbonate it puts into the bile. And again, the secretin in the bloodstream reaching the pancreas will cause the pancreas to increase how much bicarbonate is put into the pancreatic juice. Now there is something else that controls bile secretion by the liver mentioned in your textbook. It talks about how the return of bile acids to the liver is the greatest factor that promotes more bile secretion. However, it's a little unclear if they really mean production of bile or just the promotion of bile flow through all the hepatic ducts. So it doesn't really matter for us. I want you to know those two hormones. But I did look it up, and the evidence that bile salts being returned to the liver actually stimulates the production of more bile dates back to 1919 and 1959, in which it was two studies in which it was shown that bile salts help create an osmotic force that helps bile form just because there's greater flow of bile into the bile ducts. So I'm not sure really what that's all about. So I think it's more important to know about the neural control and these two hormones that control release of pancreatic juice and bile. That's the end of this lecture, and thank you very much for listening.